Welcome to the Research Like a Pro with DNA question and answer series. Today we have the question, can xDNA help verify or refute a mother-son relationship? I love this question because we don't often get to talk about xDNA, and xDNA is so interesting and it can be very useful. Let's go over the inheritance of xDNA. So males and females both have xDNA. Females receive two X chromosomes, which are sex chromosomes, chromosome number 23. So females receive an X from mom and an X from dad. And so they are female with two X's. Males receive an X from mom and a Y from dad, making them male XY. So both males and females have xDNA. Now, xDNA only recombines for females. So when a female passes on her xDNA, the X chromosomes that she has, they recombine just like the other autosomes in autosomal DNA. And the X that the female has from her mother and the X that she has from her father go through recombination before being passed down to her daughter or her son. And so the X that you have from your mother is a recombined X from both her mother and from her father. But when a male passes his X chromosome, he cannot have the X recombine with the Y because they're separate. And so they're different. They don't recombine. And the X that the male got from his mother is passed completely intact down to his daughter. And males do not pass xDNA to their sons, so only to their daughters. So remembering that an X chromosome received from a mother is recombined, while an X chromosome received from a father is not, can help us when we're doing our analysis. Now, when we're looking for xDNA matches, there's three databases to look in: 23andMe, FamilyTreeDNA, and GEDMatch. Another thing to remember about xDNA matching is that relatively few SNPs are compared when matching on the X chromosome. So that means that rather than testing every single part of the X chromosome, there are just samples tested across it, just like in autosomal DNA. And for some reason, the testing companies choose fewer SNPs to test on the X chromosome than others. So this increases the possibility for false matches and pseudo segments. So one of the recommendations is that we use segments that are a little bit larger. We don't want to use small segments because small segments have a larger possibility of being a pseudo segment. And if we stay above 15 to 20 centimorgans, then we're in a more safe zone. So how do these pseudo segments get created? Well, it's because of these females that have two X chromosomes, and usually the testing companies are not able to phase them, although some testing companies are trying to do phasing now. And phasing just means they separate the two nucleotides at each SNP into maternal and paternal. Otherwise, they don't know which one is maternal and paternal, so the algorithm can just go back and forth to whichever one matches another person's segment and then figure that out. But let's say that these are phased and that this is the maternal and this is the paternal and the same here. Then a pseudo segment would be created when the testing company algorithm doesn't know which one is maternal and paternal because they're unphased, and it tries to match the segments, but then goes up and gets the wrong letter from the other side, and then does the same with the other. And so these two would both be pseudo segments and they'd be matching, but they wouldn't be a real match. You would want to see matching on just one of the phased chromosomes with just one of the other phased chromosomes. So when you are doing xDNA matching, you don't have to worry about this at all when you're comparing two males. So if two males have an X match, there's not going to be any false matches because they only have that one X chromosome that was passed down intact from their mother that doesn't recombine with the Y. So they're not going to have pseudo segments. There's no way to create a pseudo segment when one is a Y and one is an X. Then for male to female, there can be pseudosegments created between the female's unphased X chromosomes, but the male won't have that. And so there's a little bit less of a chance of a pseudosegment being created. So we still want to use larger segments, probably over about 15 centimorgans. Then when you have two females, there's a potential for a lot of pseudosegments, like this example, where both females have these unphased two X chromosomes. And so the algorithm can be weaving back and forth between the various chromosomes and getting pseudosegments. And where there's two females, then you want to use a higher threshold for your minimum. It could be 20, maybe 30 centimorgans to reduce the chance of getting a false segment. It doesn't mean you wouldn't want to look at something that was between 15 and 20, but just don't spend as much time on it, especially if you're not finding a common ancestor. And it can be really hard to find common ancestors with X matches because the X DNA can go back quite far in our tree, and sometimes we can't find the common ancestor. So for an example, I will share an example of a son receiving xDNA from a hypothesized mother. And let's say we want to confirm or reject this relationship. Can we do that using xDNA? So the answer is, the question is, who was the mother of John Robert Dyer, born in 1813 in Tennessee? And so John Robert Dyer would have received his Y chromosome from his father and his X chromosome from his mother. The X that he got from his mother is going to be recombined from both of her parents. So it could have DNA from both her father's side and her mother's side. So the Taylor side and whatever other side. And so, yes, the X would be passing to John Robert Dyer. And if he passes that to any of his descendants, then there are any living descendants who receive that xDNA, we can possibly look for their matches. So xDNA inheritance for males comes only from his mother's side, and then the mother's father's side, a limited amount, and then more from the mother's side. And you can see that xDNA inheritance is kind of tricky, it's not too obvious, but the main thing to remember is that it does not pass through two males in a row. So you would never see a blue and a blue because remember, fathers can only pass a Y to their sons. So when we're doing our research planning, we can think about different strategies and different testing strategies we might want to do. The first strategy I thought of was descendancy research to determine which potential test takers received xDNA from John Robert Dyer, since that's our question. So we'd want to look at a big descendancy diagram. And then in our descendancy diagram, we just want to remember the tip that xDNA can't pass through two males in a row. So for example, this first one, if it passes from John Robert Dyer to one of his daughters, then to another male, then to a female, and to a female, and then to a male, that would be fine. This male has xDNA, who he received from his mother, who she got from her mother, and she got it from her father, so that's fine. What about the second one? If John Robert Dyer passes it to his daughter, who passes it to her daughter, who passes it to her son, would he then pass it to another son? No, right there, that would be a break in the xDNA, so this line wouldn't work. So we need to look at our descendancy diagram. So here's the descendancy diagram of John Robert Dyer. He received xDNA from his mother, and these are the children who I have descendants of. And so none of these green sons are going to carry any xDNA from John. They only got Ys from him. But the female daughters, so Mary and Elizabeth, did receive xDNA from their father and from their mother. And so the potential for finding matches to one of their descendants is there, so we can trace them down. So Mary had a daughter, Bathsheba, and she received xDNA, who had a daughter, Laura, who had a daughter, Pearl, who had a daughter who has shared her DNA matches with me at Ancestry. Ancestry doesn't show xDNA, so I'd have to ask her to transfer to GEDMatch or to FamilyTreeDNA. And then the daughter Elizabeth had a son named Richard, who he received xDNA from his mother, and he had two daughters, Bonnie and Maud. And Maud passed her xDNA down to her daughter, who is a test taker who has shared her results with me at Ancestry. And Sarah has, the I'll use the name Sarah. Sarah has uploaded her results to GEDMatch. So that's exciting. So I can then go to GEDMatch with Sarah and I can look at her matches that are xDNA matches. And potentially some of them could descend from John Robert Dyer and maybe Sarah Taylor. So let's look at the tree for Sarah because Sarah is not my ancestor. My husband's ancestors are on this side, the family through John James Dyer, who didn't receive xDNA. He received Y-DNA, and we've actually done Y-DNA testing on this branch. So for Sarah, these are her ancestors, and she received an X from her father and from her mother, and I put stars next to everybody who potentially passed the X chromosome down to Sarah. And this helps because when we're looking for common ancestors, we want to know which ancestors potentially contributed that X to Sarah. Then I went to GEDMatch and I put in her kit number, Sarah's kit number, and then I actually didn't choose autosomal, I chose X. And this helps because then instead of scrolling through all the autosomal matches to find who would be an X match as well, I just could see only the X matches in order from largest to smallest. So that's helpful. And so over here is the xDNA column, and I blew it up a little bit, but it basically shows you the total amount of xDNA and then the largest segment. Now, all of these 196 centimorgan X matches are Sarah's children. So I skipped past those and went down to the next highest matches. And this first one, 56.7 centimorgans on the X chromosome shared with Sarah. This is going to be match number 1, and 53.4 will be match number 2. And so one of these had a tree, and so I was able to look at that tree. So I thought this would be good to analyze. This one didn't have a tree, but I think it's a sibling to this match with the same surname. All right, so some of these matches don't share any autosomal DNA. They only share xDNA. So that shows you these are further back than second or third cousins because we share DNA with all of our second cousins and most of our second cousins once removed and third cousins. So probably further back than a second or third cousin would be these X matches. Some of the X matches do share a little bit of autosomal DNA, like this one shares 10, this one shares 5, this one shares 34. So you might prioritize looking into the ones that share autosomal DNA and xDNA, hoping that they're closer, but 34, 5, and 10, those could be just as distant. And these just happen to be the ones that you also share autosomal DNA with. I did find that match 1 and 2 all triangulated with Sarah. So the potential there is that they all share the same common ancestor that they inherited that xDNA from. So the way that I got these 3 screenshots is by clicking on the segment of xDNA. So if I click on this 56 and I click on the 53, then I can see that Sarah and match 1 share this part of the X chromosome. Sarah and match 2 share this part. And then I took the kit numbers of match 1 and 2 and compared them and found that they share the same one. So they all share this segment with each other, meaning it's triangulation. Now, since these 2 are males, you can see the match is very high confidence, all green. And that's because there isn't that possibility for, I think it's because there isn't that possibility for mismatching nucleotides at the different SNPs. So let's look at the trees of match 1. So I was able to find that the parents of match 1 were on Family Search with a full tree. So here is match 1, and he got all of his xDNA from his mother. So right away, we're able to narrow down to just this half of the family tree. And then I put this DNA symbol on all the people who could have been given him his xDNA. So we are going back on his mother's line all the way back, and then his mother's father's side on the mother's father's mother's side. So all of these different people that have the symbol could have been the ones that contributed it. Then on match 2, I built his tree on Ancestry and focused on the maternal side since all of his xDNA came from that side, from the mother's side and the mother's father's side here. And I was able to get pretty far, but I could have spent a little more time on this. It takes time to build these trees out. So once I got about this far into the building of the trees, I decided to compare surnames. So I just made a list of all the surnames that I had so far that had the X. And for Sarah, we had all these surnames. Match two had a shorter list, and match one. And I noticed Carter was an overlapping surname, but match one, the Carter surname didn't have a first name attached to it. So I needed to do more research. It was just a guess that the mother's name was Carter. One thing I did notice is that a lot of these xDNA ancestors from all three trees were from Kentucky, North Carolina, and Virginia. And so that was exciting because I know that John Robert Dyer's hypothesized mother was probably from Kentucky. And there's two counties I found, Butler and Warren County, where a lot of these Taylors came from, and they had migrated there from Craven County, North Carolina. So I am excited to continue to examine these xDNA matches, building their trees back, see what locations really stand out. But North Carolina definitely stood out as a connection in the locations. So the conclusion is that xDNA can be interesting and very helpful. And we do need to build the trees back in time because, especially when a male passes that X chromosome intact to his daughter without recombination, it's preserving that without changing any of the chromosome for a full generation without any recombination. So it's kind of like reducing the number of generations that you would normally think of, like with autosomal DNA, where there is that recombination in every generation. So whenever it goes through a male, then you're basically extending the pedigree back even further to where the common ancestor could be twice as far back in the tree, which can be really helpful, but also can be challenging to find. So we have to build the trees. One thing to do with the xDNA is to really use the shared matches tool. And I didn't show that, but I did use the GEDMatch shared matches tool, which is called people who share with both of these kits, something like that. And so if you put in the kits of the two people who are X matches, you can then look and see who their matches in common are. And sometimes with those people's trees, or if you recognize their names, you can see which side of the family they're on. So that can help narrow down the X matches even more. And what I found is that we really just need to log everyone we look at in the research log and any discoveries we make. And there's a lot of clues in GEDMatch, a lot of links to different tools and people's profiles and their wiki trees and all these things that can help us find the answers, but we have to log them because it's hard to keep track of them all. And I ended up having so many browser tabs open as I was working on this and finally realized, okay, it's time to really just log everything I'm doing here in my Airtable research log. So hopefully that gave you a taste of how xDNA can be used as we're trying to verify a mother-son relationship. And obviously I didn't get to an answer here. It's going to take more time. But it is exciting to work with xDNA and it can be really a fun thing to try and work on. So good luck.